Okay, thank you for the introduction. So, good morning everyone. Today I will talk about resistance to Orobanche cumana in sunflower, and I took some lyrics from the famous lyrics from Queen Ben. And but I'm not sure that one day we will never. I mean, the story I think will never end. So we will illustrate it. But first, before starting, I would like to present my research group. We are eight people, 4 permanent people: Mireille Chabot, Marie-Claude Boniface, Alexandra Legendre, and myself. And we have complementary skills. And there are also students. And Camille Pubert is attending the meeting today. She is performing a PhD in collaboration with Masseeds and on genetics, on the cloning of the HR-OR5 gene, and I will present it later. And they are my Wonder Woman. I mean, without them, nothing could have been possible. And I really thank you, Mireille, Marie-Claude, and Camille, who are taking the meeting. So, Orobanche cumana is an obligatory parasitic plant because it is non-photosynthetic. It does not develop any roots. It looks like an asparagus, as you can see there. And using a susceptible control line 2603 without any bloom rate, and then compared to the same line inoculated with Orobanche cumana, you can see a clear decrease of the growth of the sunflower and delay of the flowering. And we have also a strong effect on the number of seeds per capitula. So in the field, the Orobanche cumana can induce very strong yield losses. The cumana seeds are very small. They look like powder, as you can see here in a box we collected cumana seeds. And only one plant can produce 100,000 seeds, and the seeds can survive more than 10 years in the soil. So they can rapidly spread from one field to another. The biological cycle of Orobanche cumana is mainly divided in 4 steps. The 3 first steps occur underground. So the first step, the seeds located in the soil, we perceive a stimulant, germination stimulant from the sunflower root exudate, so they will germinate. Then they will penetrate the sunflower root and connect to the vascular system. Then they will develop a storage organ, a tubercle, and then a flowering shoot will emerge above ground and produce the new seeds. So the first report of Ocumana was in Russia in 1866, and then Ocumana spread around the countries around the Black Sea, and then in Spain, and we found recently Ocumana in France and also in Israel and in North Africa. So the population, the worldwide population, differ from their level of virulence, but also their aggressivity. And we have a very similar situation than for downy mildew, and their virulence are named from A for the less virulent population to H for the most virulent population. And later, Molino Ruiz proposed, I think, something very interesting, using triplets of differential lines, like for downy mildew, and a new nomenclature of the different races. So the situation for the race is clear up to E, but for more virulent population, it's clearly unclear. We have race S, S plus, G, G plus, G from Guadalquivir, H, and so on. So we really need now a universal set of differential sunflower genotype to differentiate the different races. Concerning the molecular diversity of the population, so Lilian Liu from Zhejiang University came 2 years in my group, and in collaboration with Inovea, we did diversity analysis, and we found 5 different genetic pools one corresponding to the Chinese population, one to the Eastern Europe population, one from the French population, and two genetic pool in Spain, one from the population in Guadalquivir Valley and the other in the Cuenca region. Concerning Orobanche cumana genomic, we sequenced the 19 chromosomes of Orobanche cumana genome, and we obtained the full genome sequence of 1.47 gigabases of the IN23 population. It is an homozygous population produced by CSIC in Spain, and it corresponds to race F from Guadalquivir. We annotated this genome, and we found more than 43,000 genes coding for protein. And more recently, we also obtained very high quality genome of 4 new races from other countries. And we also resequenced using Illumina sequencing data the genomes of 21 population, 6 from China, 3 from France, 5 from Spain, and 7 from Eastern Europe. And we found more than 5 million SNPs. And we now want to understand the evolution of the population and try to link the polymorphisms with the virulence of the population. So now I will go into how to ask the question, to respond to the question how to control Orobanche cumana, and the first method to control cumana is our genetic resistances. So we mapped quantitative resistance in this paper. But we found many QTL specific to the different stages of the interaction, but none of them gave a complete resistance. Concerning the major resistant genes, so there were the first one was OR5, and it has been located in the upper part of chromosome 3. And there is also more recently a major gene. But inducing post-osteroid resistance, inducing phenolic compounds, necrotic tubercle, and reducing the growth of Orobanche cumana, and the name of the gene is system. So now I will detail our work on the identification of the first resistant genes for Orobanche cumana. It is the name OR7. So it was done in collaboration between INRAE and Syngenta, and it was the work of Pauline Duriez, my former PhD student. She is now a postdoc at UBC in Lorraine Heisberg Lab, and I have an announcement. If you have a job position for her, she's looking for a position. Okay? So OR7 was first introduced in a pioneer hybrid in the early 2000s, and this gene provided a total resistance against race F without any emergencies of cumana in field. And it is interacting with an avirulent gene in a gene-for-gene interaction. But we did not have any information about nor the physiological mechanism of the gene, nor the identification of the gene. That's why we wanted to identify the gene. So first, we used GWAS approach using a set of 55 lines, and we genotype, Syngenta genotype, this time using 609K SNP action sheet, and they found a clear location of the gene on chromosome 7. They confirmed the location of the gene using a small F2 population of 355 F2. They genotyped them and phenotyped them and mapped the gene on chromosome 7 in a 1 centimorgan region corresponding to 850 kb genomic region. So we wanted to identify the gene. That's why we now screen it by genotyping a very large F2 population of more than 14,000 F2 plants. And we phenotype them, the recombinant plants we identified during 2 years in south of Spain. And finally, we located the gene in a 55 kb window with only one gene encoding for a leucine-rich repeat receptor-like kinase. So OR7 is encoded by this gene, and this gene showed homology with a resistant gene conferring resistance to Xanthomona oryzae in rats. We also sequence using backclones because at that time, it is a long time ago, it was not so easy to sequence the genomes. So we use backclone strategy, and we sequence the genomic region of the resistant line and the susceptible line. And when you have a black line, that means that there is homology between the two lines. Okay. So as you can see, there are only homology for the genes, and no homology at all in the intergenic region, meaning that first the OR7 has a wild origin, and now we never found the origin. Even we produce many wild relative and wild annulus genomes now, but I didn't find the origin. So if someone from Corteva could give me the origin, I will be very happy, you know. And we found also that in the resistant line, the leucine-rich repeat receptor-like kinase was duplicated. So finally, we also describe the physiological characterization of the resistance, and as you can see, like in the resistant control, the resistant line produce only incompatible attachments, and like in the susceptible control, the susceptible line mainly produce compatible attachments. And we found also that all the susceptible lines encoded for a truncated protein with a stop codon, and in this susceptible line, the protein was lacking the transmembrane domain and the intracellular kinase domain, while all the resistant line encoded for a fully functional protein. So we propose that this kinase can interact with the navirulent gene from cumana, and then induce cellular transduction that prevents the connection of cumana to the vascular system. So if you want more detail, this work has been published in Nature Plants in 2019, and now we have a new project for the identification of the navirulent gene interacting with OR7 in collaboration with Syngenta, my group, and the group of Begonia Perez-Vic and Leonardo Velasco in CC. So now I will talk about another gene, very important. I think the best gene today we have to use to fight against cumana. So it was they used Nelanthus debilis, tardifolius, to introduce in cultivated annulus a resistant gene conferring resistance to the highly virulent race gene. And the gene was monogenic and dominant, and the mapping of the gene has been recently published in theoretical and applied genetics. And now, Belén Fernandez, the PhD student supervised by Begonia Perez-Vic in C6, came 4 months in my group, and we sequenced the DEP2 line genome. So as you can see there, you have the matrix of the genome. So it is a very high quality genome. And we also, in the frame of the International Consortium on Sunflower Genomics, we also sequenced another Nelanthus debilis genome, not the one used for DEP2, but as you can see there is no homology between the DEP2 region and the XRQ, the cultivated XRQ region, but there is a high homology between the DEP2 region and the Nelanthus debilis region. So now Belén is working on the identification of the candidate genes and to identify the resistant gene DEP2. Okay. So now I will talk about the cloning of the HR-OR5 gene, because I think it could be a model to better understand the molecular mechanism of the interaction. Because the avirulent gene interacting with OR5 has been already identified by the colleague from CC. So it is the work of Camille Hubert, and we used a similar approach than for OR7, and the gene was mapped at the beginning in a 1.5 megabase region. And now, after screening a large F2 population and phenotyping the recombinant plants, Camille succeed to restrict the location of the gene in a 193 kb region. We sequence the susceptible line and the LC1093 resistant line we used to map the OR5 gene. And as you can see, the metrics are very similar and the quality of the genome are very good. And in fact, we compare the susceptible line there and the resistant line LC1093 and as you can see, we found a very large insertion in the resistant line that could contain more than 200 kb, and this very large insertion could contain the resistant gene. So finally, what we want now, we are very close. We identify the first resistant gene. We are very close to identify new resistant genes, but we want to validate this resistant gene using sunflower transformation. And Mireille Chabot, my group, developed root transformation using agrobacterium rhizogenase. As you can see there, we can so she transformed the plants using the GFP protein. So we clearly differentiate the transformed roots that are fluorescent, and she then inoculate these roots with Ocumana, and she succeeded to inoculate the transformed root with Ocumana. And here you have a cross-section of the root, so you can clearly distinguish the fluorescent cells from sunflower from the one of Orobanche cumana. Okay, so it will be a very useful tool to validate the resistant gene in the future. So now I will talk about new control methods. It's another way to control Orobanche cumana. So we are now, I will talk about a new project that is named STIGO. We are obtaining a funding of more than 700,000 euros from the French National Agency for Research, and 5 different partners participate to this project. I am the coordinator of this project. So the CNRS from Gilles-sur-Yvette, INRAE from Versailles, the Nantes University, the company Inolea, and my group, my lab, sorry. And there, in this project, there are 3 main objectives: to the identification of the Ocumana seed germination stimulant and their biosynthesis genes in sunflower using different approaches, the characterization of the receptor of this stimulant, and finally to develop new control methods. I mean, to try to develop sunflower that do not induce the germination of Orobanche cumana, but also maybe to, in more long term, to have a new agrochemical control of Orobanche cumana using the results we will obtain. Another project that was coordinated by Philippe Delabo was the use of the micropeptides. So micropeptides are very small peptides of about 10 to 20 amino acids that can be synthesized or produced in biotanks by using bacteria containing these micropeptides. So using both the sunflower and the cumana genomes, we identified different micropeptides, and Philippe will talk about that in few minutes. Another project is the Homoco project in collaboration with Marcis, Agir, Celia Seasso, and my group. So in this project, we wanted to assess the winter crop and biofumigation as a control of sunflower movement. Okay? And for that, we did experiment in control condition. We used hermetic containers, okay? And we use, so for example, we had control without cover crop, so only cumana seeds, and in another box we put grinded cover crops tissues, okay? And then this cover crop will liberate some volatiles like the isotosana or the secondary metabolite, and we wanted to know if they could have an effect on Ocumana seed germination. So we counted the germination rate. And I was very impressed. So here you have the control without cover crop, okay? And here you have the result of with one species of cover crop, and this cover crop clearly inhibits the germination of cumana at 100%. So I was very surprised, so I asked the student to repeat the experiment several times, and we found the similar results. So this is a way to control very well the cumana. So finally, I will thank all the collaborators, and mainly I started my first project on cumana in 2013, building a very strong collaborative network with Nantes University. So Philippe Delaveau, I want to thank you so much for your help and for all the projects we have. And also Cécile qui est in Cordoba, so Leo and Begonia, thank you so much for your help, for all the projects we had and all the results we obtained. Thank you so much.